We are going to go through topic 3, DNA. So this is starting on page 30 of your workbooks. And here we go. So before the compilation of Human Genome Project, many scientists were expecting to find 100,000 or more genes in our genome. This was based on the assumption that because we are one of the most complex creatures on Earth, we should have lots of genes. However, it turned out that we only have about 24,000 genes. So the genetic information of a cell is contained in what's called DNA in the nucleus. So we already talked about DNA and how it's important to cell division, but pretty much the root of it is DNA makes all of our information in our bodies and who we are. So DNA is unique to each individual. It's different for everybody. Even though we're supposed to all have the same type of genes, just like we said in topic 1 and 2, alleles are what makes it different. So all DNA is different for everyone. All right, so DNA stands for deoxyribonucleic acid. So D for deoxyribo, N for nucleic, and A for acid. The deoxyribose is the sugar that makes up the backbone, so the sides. So if we look at this DNA structure right here, this is the sides. On each side, there's a red one and a blue one. Those are your deoxyribose backbones. The name nucleic is from it being found in the nucleus. And the reason it's an acid is because there's these little proteins also in the backbone that are called phosphates. And these are the backbone, also creates the backbone of the DNA, and it makes it acidic as well. So DNA is universal, meaning the code is all the same. It is a genetic code for all living cells, whether they are plants, animals, bacteria, or fungus. Okay, so where is DNA found? So there are 3 places that DNA can be found. 2 of them are going to be in animals. 1 of them is going to be in plants. The first one being nuclear DNA. So the nuclear DNA is found in the nucleus. Okay, right in the name. And we already talked about this in topic 1 and 2. And this is where we find long strands of chromatin. Remember, chromatin is DNA wrapped around histone proteins, and then it condenses to become chromosomes. We can also find it in the mitochondria. So even though the mitochondria is known for the powerhouse of the cell, it also contains DNA as well. So the DNA here looks a little different than the ones you find in nucleus. It's more circular in terms of its shape. And with mitochondrial DNA, we'll talk about this in the next slide, it actually comes from your mom, or your maternal genetic information gets passed down from your mother. Also, the last place where we can find DNA is found in only plants. So this is called chloroplast DNA, and the shape of it is also circular. Remember the chloroplast, if you remember back in Science 10, that is where you can find chlorophyll, and that's what makes plants green. So mitochondrial DNA and chloroplast DNA is used to study the evolutionary relationships between species, so between animals and plants, and they're much smaller than nuclear DNA, so they're much easier to look at and to dissect. So like I mentioned, in humans, an individual's mitochondrial DNA is identical to their mother's. So nuclear DNA comes from both parents, whereas mitochondrial DNA, that's going to be passed down only from your mom. So if there was some sort of disease that's found, or genetic mutation found in the mitochondrial DNA, only the females in that family can pass it down. So on the left side, we have nuclear DNA here. That means you're getting DNA from both mom and dad. So mom and dad. And then here on the right side, mitochondrial DNA only comes from your mom, your maternal lineage. So only the females. Notice how only the females can pass it on. If it gets passed on to a male, he does not pass it on. Okay. All right. let's talk about how DNA is structured. So Chargaff's rule indicates that A, adenine, is equal to T, thymine. Okay, and then G and C are also equal. So guanine and cytosine are also equal. Rosalind Franklin, so let's just go back a little bit. So Rosalind Franklin was actually the first person to determine what DNA looks like. So she used X-ray diffraction to get the structure and to study DNA itself. So she came up with the idea that DNA is a helix and it is double-stranded, and that the distance between the DNA strands are constant or the same. So if you look at this DNA structure right here, a helix means that it is twisted within itself, and then it's double-stranded, so there's two sides that are twisting, kind of like a ladder around itself. So from this, two other scientists, Watson and Crick, came up with the 3D model of DNA. Okay. So again, Watson and Crick determined the 3D model structure, so they actually brought it to life from the study of Rosalind Franklin's work. So just a little bit of history here. Watson entered university at 15 in Chicago. He was at Cambridge University, where he met Crick, who was a physicist, and together, with the help of Rosalind Franklin's work, they came up with what the DNA structure looked like. Okay, so a little bit more in detail of the structures. So we'll go back. So DNA looks kind of like a ladder, like a twisted ladder. So if you think of like, those really big houses with those grand bougie staircases that just come down the middle of their house, think of it like that. So in this case, it just looks like a twisted ladder. So the structure of it, we have what's called two complementary strands. So each side of the DNA are partners, essentially. They're not the same. So if we think back to Chargaff's rule, if we have something called A, or adenine, its partner is going to be T, or thymine. Same thing with C, cytosine, is going to be partners with guanine, or G. We'll talk more about what those letters mean in a second. But essentially, those are going to partner up and make complementary strands. So not the same, but partners. We have the backbones. So here it's highlighted in the red. We have the backbones, so the sides of this, the DNA strand. And remember at the beginning we said that it's made up of deoxyribose and phosphate. So think of the backbone as the staircase's handlebars. As you're going down the staircase, the backbone is the handlebars that you're holding on to. We have what are the rungs? So the middle parts of your DNA strand. So the rungs are like the stairs itself. Okay. And in this case, we have what are called nitrogen bases. So nitrogen bases are what make up our code, or the coding in a DNA strand. And this is where that A, T, G, C comes into play. So these four are our nitrogen bases in DNA. So adenine is a nitrogen base, again, that pairs with thymine. So A with T, guanine pairs with cytosine, G with C. An easy way to remember this is that all the straight letters go together and all the curvy letters will go together. Or you can think of apples grow on trees, so A and T. The carrots grow in the ground, so C and G. Okay. Now, you notice in the middle we have these dotted lines. That means that is the bond that's bringing the two nitrogen bases together, or holding them together. And those are hydrogen bonds, is what's going to bring them together. Okay, so our structure, just to review, we have the backbones, the sides that are made up of deoxyribose, which is the sugar, and phosphates, which is also part of the backbone. In the middle, think of like the stairs on a staircase. Partnered up, we have what are called nitrogen bases. And remember, adenine, A, goes with thymine, T. Guanine, G, goes with cytosine, C. And then together, those are all held by hydrogen bonds. Okay, so make sure you guys know all the different structures. Okay, awesome. So in this case, we can figure out the complementary strand if we're given one side of the DNA strand. So remember, complementary means that it's its partner, not that it's identical or the same. So if we're given this strand of ATC, GAA, TCG, we can find the opposite, or not the opposite, the partner strand of it by finding the partners that they go with. So remember, A goes with T and C goes with G. Awesome. So if we have ATC, it's going to become TAG. And then GAA is going to become CTT. And then TCG is going to become AGC. Okay, super simple like that. Okay, so when we're looking at the structure of DNA, we have to look at the what makes up one unit of it. Okay, so we have something called a nucleotide. So the functional unit of DNA is called a nucleotide. And what a nucleotide is if we just zoom in or look at one section of the DNA strand, one nucleotide is made up of one sugar or deoxyribose, one phosphate, and one nitrogen base. So in this case, there's three things that make up one nucleotide. Okay. DNA is made up of many nucleotide monomers. Monomers just means one unit of it or one structure of it. So in this case, if we look at this example, okay, we have this DNA strand. Here, this is one nucleotide. Here, this is the second. Here, this is the third, the fourth. And we look at both sides, so 5th, 6th, 7th, and 8th. So here we have 8 nucleotides. We have 4 on each side. Okay. So again, this is what a nucleotide looks like. It's made up of one phosphate, one deoxyribose or one sugar, and one nitrogen base. And that can be either A, T, C, or G. Okay, here's a little GIF. So nucleotide structure, one sugar, one phosphate, and one nitrogen base. Okay, so here's a practice problem you can do in your workbook where you're going to label these structure names to the diagram. Okay, so when you're ready to do that, you can pause the video, and when you're ready to come back, play again. Here's the answer for it. Okay, so here you go. Here, this is your phosphate. The little pentagon, that's your deoxyribose or sugar. In the middle, one of them is your nitrogen base. And then in the middle, between the nitrogen bases, you have your hydrogen bond. And then remember, one of each, so a sugar, a nitrogen base, and a phosphate together make one nucleotide. Good. Okay. Awesome. So your DNA structure, remember the different stages of DNA. You have your